Hello students, this recording is for Biology 1407 lecture, and it is Chapter 19, the second lecture in Unit 1 on evolution. Chapter 19, the evolution of populations. So I want to walk through the lecture content right here, and this is of course the OpenStax Biology 2nd edition textbook that we're using as our reference. Alright. So, the subject is population genetics, and we're going to go over some material in here that is extremely useful for you in in laboratory, your ALU transposon experiment from laboratory. This is where we're going to also utilize the Hardy-Weinberg equilibrium, which we're going to talk about in just a moment. So the subject matter is, for the laboratory and for lecture here, is population genetics. So, the genetics about the gene pools, a discussion about the gene pool of a population. And remember that the definition of a population is individuals of a particular species, typically that are constrained in a particular geographic region or, some other sort of portion of the environment that's out there, and members of that population have exchange of genetic material amongst the population. In other words, mates can be found amongst that population. Another population is by definition isolated in some degree from a different population. The most obvious example of this is think of freshwater fish in lakes. Each lake has its own population of a given species, and the gene pools of those populations can differ pretty substantially. And of course, from Chapter 18, we learned that substantial change of gene pools between two populations of the same species can lead to speciation by preventing reproductive compatibility when those gene pools are brought in contact with each other again, when individuals from one population make contact with individuals of the other population. If they won't mate or can't mate, for whatever reasons, the gene pools are isolated, and effectively they are new populations. Once they become reproductively incompatible, by definition they are different species. So that's the subject of population genetics. All right, so a little terminology right here. In Chapter 18, we started by looking at Darwin's work and the other naturalists' work that was drawn from observational studies in the wild, in the field mostly, collecting animals and plants and fossils and whatnot, looking at things, coming back, examining them, dissecting them, and building a terrific database back in the early 1800s of a detailed analysis of the diversity of life. That was one portion of evolutionary work. The other came from a more experimental side, and one of the first people that really contributes substantially to our modern understanding of evolution is Mendel, famous for his quantitative experiments with inheritance, patterns of inheritance in pea plants, garden peas, and other plants, the Austrian monk Gregor Mendel. Now, he didn't really work on evolution. That's not what he was interested in. He was interested in heritability of traits. And so he did really, really excellent, rock-solid, experimental, manipulative science that had a numerical underpinning and was could be used, statistical analyses and various other mathematical tools could be used to analyze that data. In the 1940s, we put those two pieces of work together, along with some new information coming from an investigator named Morgan and his work in the heritability and patterns of heritability of mutations in Drosophila, the fruit fly, and his work was directly spurred by the work of Mendel in the century previous. Putting that experimental quantitative heritability work together with Darwin and Wallace's and other experimenters' work on natural selection is the modern synthesis of evolution. So where we distinctly put heritability and the quantified, understanding of heritability together with evolutionary theory based on natural selection. So, using this language, we have a gene pool, and of course a gene pool is all of the genes that code for all of the effects, RNA, messenger RNA, proteins, depending on the particular gene, for a given species. And the gene pool includes all the different alleles, the versions of those genes, blonde hair versus brown hair or black hair, and the frequencies, how many copies of a particular allele are out there in the population. Some things will be relatively rare. There is an allele for one of the pigment genes of the 3 sets of genes that are responsible for our body pigments that has a mutant allele version that's responsible for albinism. That is relatively rare. It's a rare mutation. It is distributed in the human population amongst all ethnic groups, but it's a relatively rare mutation. And it's one that tends to show up. In other words, the mutation has appeared again and again and again, probably because of a weakness, a tendency, a propensity for a mutation in a particular critical spot on this gene. And that's the sort of stuff we're talking about when we talk about allele frequencies in a gene pool. So evolution is a change in those allele frequencies over time in a population. That's what microevolution is. Macroevolution is what happens when we isolate these populations from each other and they go down different evolutionary trajectories, speciation. Macroevolution is lots and lots of microevolution leading to speciation. All right, so we want to talk about Hardy-Weinberg for a while, and as you'll recall from my classroom recording this past Wednesday the 27th, we discussed in laboratory, or I talked to y'all a little bit about why we needed to use this Hardy-Weinberg principle, what it established, and how we could use it in a laboratory setting, which is what we're going to be doing and what we are doing in laboratory with the ALU transposon experiment. So hopefully you'll recall that the Hardy-Weinberg equilibrium is a mathematical tool that predicts the frequencies of alleles for a gene. It only looks at one particular gene. It's not the entire gene pool for everything, hair color, digestive enzymes, growth factors, etc., all the stuff that makes us up, but just one particular gene. And Hardy-Weinberg equilibrium is a mathematical prediction of the frequency of alleles for that gene in a population given an initial distribution. And the idea of the Hardy-Weinberg equilibrium is if we can identify that a gene and its alleles are in Hardy-Weinberg equilibrium, it requires 2 alleles, which is a very typical situation. We have lots and lots of genes that have 2 alleles. There's plenty that have more, but Hardy-Weinberg, as we present it right here, only works with 2 allele systems. And the general idea is that there is a dominant or a more common allele out there, the one that's most frequently found in a population, and then another allele that is rarer or less frequent in the population. And for Hardy-Weinberg equilibrium to be maintained, and the prediction is that allele frequencies won't change over time, except due to the effect of random selection, which is an effect of small sample size. The allele frequencies, whatever you have in one generation, will be maintained for generation after generation. Therefore, a common version of a gene will continue to be common in generations in the future, and a rarer version of that gene will also continue to be rare in future generations of that species if a series of assumptions are maintained. The first one is that the population is large. If you have a small population, random variability can overwhelm a pattern. I tried to talk to you all about coin tosses in our last live meeting, and if you toss a coin once or twice, it's doubtful that you're going to get the typical 50-50. Well, if you toss a coin once, you don't get the 50-50 ratio of heads and tails. And if you toss a coin 2 times or 3 times or 4 times, let's just do even numbers, 2 or 4 or 6 or 8, you know that those small samples, especially of 2 or 4, are probably going to be unbalanced. 2 heads, no tails. 3 heads, 1 tail. If we do 6, 4 tails, 2 heads. Those are not unusual circumstances. But if we keep doing those 4 samples over and over and over again and average out the results, we'll see eventually that we have pretty much a 50-50 ratio of heads versus tails. Or if we just throw 1,000 coins or 10,000 coins or 100,000 coins, we'll get very close to that 50-50 ratio. That's the large sample size question. So when a population is very small, it's really easy for a rare allele to vanish because it just by random chance, it wasn't copied. Nobody mated successfully and created an offspring using that allele. Not because there was anything wrong with it, simply because it was rare. And if you don't have many attempts to do something, a rare thing, you're very unlikely to see that rare thing. A very large population has a high probability of using that allele in a successful reproductive event, and the allele therefore persists in the population. So sample size is a key feature right here. And then we had other assumptions. There is no natural selection. One allele is not better than another. So in this particular case right here, looking at Mendel's pea plants, the yellow pea seeds and the green pea seeds right here, neither is better or worse in this particular, that's the hypothesis. Whether that's true or not depends on experimentation. But the analysis allows us to kind of determine if there's something going on. So no natural selection, no sexual selection. So this is plants. Their sexual selection requires the action of, a third party, an insect or other pollinating entity, for sexual selection to be at work right here. But the assumption is there's no sexual selection. So the likelihood, based on whatever the reproductive strategy is, has no favoring between yellow versus green seeds. That's reproductive, that's neutral or lack of sexual selection. There is no genetic drift. There is no genetic flow. No individuals are leaving the population taking copies of a rare allele or a common allele away and therefore changing the allele frequencies, and no new copies of one or the other allele are entering the population. So we're not changing the ratios through immigration or emigration. So if all of those things are true, then we have this hypothesis that these ratios will be, remain constant over time. So the flip side is you can do an experiment and test this question. And if we find that the ratios don't match Hardy-Weinberg equilibriums, that what we're seeing in the population is not maintained over time, but instead changes, then one of those assumptions is being broken. Either the population is too small, natural selection is having an effect, sexual selection is having an effect, genetic flow, the movement of individuals in or out of the population is at work. These sorts of things are going on. And if that's, what Hardy-Weinberg tells you is that it doesn't tell you which of those assumptions is being violated, but one of them is. And if we can remove the population size question from it, then we can really say that the forces of evolution are at work on a population. If these ratios are not what the Hardy-Weinberg equilibrium propose. That's the power of this tool. So it tests the null hypothesis of an experiment. And if you reject the null hypothesis, you have to accept the alternative hypothesis. These alleles are under some sort of selective pressure, whatever it may be. Or the population is too small, or there's genetic flow, gene flow. So that's the idea, all right? So how do we do this work? Well, we take a look at a population and we recognize that all of the genotypes, here's homozygous for one allele, heterozygous, homozygous for the other allele. So this is just like the ALU situation. If we look at the proportions, and if for here we'll just assume there's 100 individuals just to make it, 1,000 individuals just to make it really neat and tidy right here, the proportions, must add up to 1. So 245 individuals right here, it is 500 individuals. what am I doing? 1,000. 245 individuals, 210 individuals, and 45 individuals. If we add this all together, that is 400. That is 500 individuals. And 45 is 0.09 of 500. 210 is 0.42 of 500. 245 is 0.49 of 500. All right, so that's what's going on here. So we've got the frequency of these alleles, and we actually have the number of individuals with these alleles. So the way a Hardy-Weinberg calculation goes is you take this data from a population. This could be the results of our ALU experiments in lab. It will be, actually. And we add up all the copies of the different alleles. So you recognize that 245 individuals have 2 times that copies of this yellow allele right here. So that's going to be 490, and there's the 490. These 210 heterozygotes over here have 210 copies of the yellow allele, the big Y right here. So that's where that 210 comes from, and that adds up to 700. So we've got 700 copies of the big Y allele, the yellow seed coat allele. And similarly, if we look at the homozygotes for green, there's 45 individuals here, so there must be 90 copies of the green allele, and then 210 individuals that are heterozygotes, so another 210 copies of the green allele in these heterozygotes, and yellow is dominant and green is recessive in this particular case. There's no dominant or recessive in your ALU situation. But we've got 300 copies of the green allele. 300 plus 700 is 1,000 copies of the allele, and that makes sense. If we've got 500 individuals, we must have 1,000 alleles for the gene coding for seed color in diploid organisms. Now, then we take the 700 and divide it into 1,000 to get a proportion. The first allele, the proportion is 70%, or 0.7. Remember, 70% and 0.7 are the same thing. They mean the same thing mathematically, right? 50% and 0.5 are both 1/2. Okay, same thing for our 300 here. We take our 300 divided into 1,000 and we get 0.3. So now we have the ratio of the two alleles, 0.7 to 0.3, and of course 0.7 plus 0.3 equals 1, or 100%, or everything. And that's a mathematical check you always do when doing these kinds of calculations. Identify how many individuals you're looking at. Once you count alleles, you should have twice as many alleles as the individuals in the population. And the individuals in the population should add up, to your total here. Otherwise, you've made some mistake in counting. Your alleles should add up to twice the number of individuals. If they don't, you've made some mistake somewhere. Do the math again. And then finally, when we convert these into proportions, these proportions must add up to 1. If they don't, you've made a mistake. Go back and start again and make sure you get your math. It's just arithmetic, but get it right. Then you'll remember the Punnett square from back in Biology 1406. So this allows us to kind of recognize why we have this formula, which says P squared. That's individuals that are homozygous for the dominant allele, in this particular case, for the yellow allele. That's the P squared people, because they've got 2 copies. They don't just have P, they have 2 copies of P, or P squared, right? 5 times 5 is 5 squared. 5 times 5 is 25. 5 squared is 25, all right? So P squared right here. And we know it's 0.49 of the total population. So we say P squared is 0.49. Now we know that PQ, the heterozygotes, there's 2 ways we get those according to the Punnett square. Remember? Remember that? So, a quarter to 1/2 to 1/4 of the 2 different genotypes. So we do PQ, but we have 2 different versions of them. So PQ we recognize is, 0.1 of this population. How? Well, that's 0.3 times 0.7. So 0.3 times 0.7 is going to be 0.21. And if we do that again, here's 0.21 over here, and those happen to add up to our 0.42 for all of the heterozygotes out there. So there's our check. The numbers are all agreeing. And then finally, 0.3 times 0.3 equals 0.09, and that's our homozygous for the other allele. In the case of these pea plants here, it is a recessive allele, so these are the green individuals. These are yellow, and these are yellow. But those are our 3 different genotypic frequencies. All right, so 0.9 plus 0.42 plus 0.09, and those should add up again to 1. If they don't, you go back and check your math. That's the predicted frequencies of these populations. Then we're going to take And so we've calculated it. We've created this supposed, what Hardy-Weinberg equilibrium says these two alleles, how they should be distributed in the population based on their frequencies, should be these ratios. So then we look at any other population, or even this population right here that we gathered the data from, and run the total number of individuals, 500 individuals times 0.49, 500 individuals times 0.42, 500 individuals 0.9. All right. In this particular case, what we'll find is that our predicted number of individuals is exactly the same because of these numbers as our observed number right here, and so we are in Hardy-Weinberg equilibrium. But you can get the exact same ratio right here of alleles, 0.7 to 0.3, with different numbers right here. There's more than one mathematical solution to these numbers that will get, derived, that will get you these two ratios right here. And then we would run through our calculations, come up with our predicted frequencies, and find that these predicted frequencies are still going to, if we've got 0.7 and 0.3, but due to a different ratio, far fewer heterozygotes right here, a lot more homozygotes here, and a lot more homozygotes over here, and just a few here, but still ultimately 0.7 and 0.3, 700 alleles of this and 300 alleles of that. It's possible mathematically with 500 individuals. And when we run these predictions right here, we'll find that our population is not, at 0.49 and 0.42 and 0.09. It's something else. The numbers, the predicted values, and the observed values don't match each other. I'll show y'all an example to make that a bit more real. All right, so I've pulled up an Excel worksheet right here that I'll post in our document so you have access to it as well. It's a calculating tool that I just set up. This is something that you can make in Excel. There's a bunch of little formulas in here. Anyway, we're going to look at an example from data that would look like something you would get in the laboratory, since your laboratory work is going to be on the ALU transposon. All right. So here's an electrophoretic gel image that I had access to. I actually pulled it off of a website. And there's a few things on the gel here, but this is the student results, for an experiment in a laboratory. And the gel has holes up here at the top. It's always convention that when you show an electrophoretic gel, especially in a teaching situation right here, the origin of the material that moved is always up at the top. The vertical columns are called lanes because the DNA moves down that lane, and it's drawn by an electrical current. And the idea here is that small things that are pulled, with a given force, a given electromechanical force in this particular case, will move faster in a given amount of time. Not further, but they'll move faster in a given amount of time and therefore go further, while larger things, this represents bigger pieces of DNA, pulled by the same electromagnetic force, are going to move a slower distance in a given amount of, they're going to move slower and therefore cover a shorter distance in a given amount of time. So electrophoresis lets you sort out particles based on their relative sizes, with the smallest particles moving the fastest and the largest particles moving the slowest. What we've got on the left and right in both cases here is what's called a DNA ladder, and it's simply a mixture of chunks of DNA whose sizes are known. 3 of them are marked right here. This big line right here is 731 base pairs. That's 731 A's, G's, T's, C's in a row, and the complementary, inverse on the other side of the DNA molecule. And this one's 416 base pairs long, and this one's, probably this one right here, is 44 base pairs long. Similarly, we've got it's just another version, another marker right here, that has a different division, sort of, each of these is supposedly 100 base pairs greater than its predecessor. Not exactly, but close. Over here, we've got a slightly different breakup, but 121 base pairs. Notice it moved, something's not quite right. Yeah, one of these numbers can't be correct. This must be 144 right here. Yeah, there must be a 1 missing right here. Because here's 926, heavier, and a little bit below it we've got 731. Somewhat below that, we've got 416, but even a little further down, we've got 383, and then way on down, 121. Yeah, this should be 141 or something, or 241. I can't quite tell. It's a logarithmic scale, by the way. The physics indicates, it insists that these things move in a logarithmically separated sort of scale right here, but that doesn't matter. Anyway, that's how you read this gel. So these are ladders. These are like rulers that have been put down, so we know the rough size of this piece. It's close to 383 base pairs long, and these pieces over here about 730 base pairs, and that's the important detail. Anyway, as you recognize, here is copied DNA that's small, that is missing the ALU insertion. They don't have it on either chromosome. This individual right here is a heterozygote. They've got it on one chromosome. The plus represents this line, bigger and heavier because there's more material in it. And this is, millions of copies of that piece of DNA, the result of polymerase chain reaction copying. And here we have a whole bunch of copies of the smaller ALU-free insertion, just like the homozygous for no ALU insertion person. And then finally the heterozygote for the ALU insertion. All of their copies are in this bucket because both chromosomes have the additional piece of DNA in that location. And there it is. So here's some results. So imagine we did this for 160 students. And we got these numbers right here. 21 of those individuals were homozygous for no insertion. 77 of those individuals were heterozygotes, this right here. And 62 of those individuals were homozygotes for the insertion right here. Well, 21 plus 77 plus 62 equals 160. I just put a little formula in here, sum of these three numbers, so it's counting them. All right, we'll see, we can use this in a little bit. And then we can go ahead and start calculating our Hardy-Weinberg terms from this. So we need to know the number of P alleles, that's this allele, or the negative allele. We need to know the number of Q alleles, the positive allele, using ALU terminology right here. And that's easy. It's 2 times this plus this. So it's 21 twice, because there's all 21 of these people have 2 copies of this allele. So that's 42 plus 77. Well, 40 plus 70 is 110, and plus 7 and 1 is 100, and 19. Yeah, sorry, 42 plus 77 is 119. I couldn't do the math in my own head for a minute. And similarly over here, 62 twice is 124, plus 77 is 194, plus 7 is 100 and 201, and there we are, 201. All right? And then and that's 320 total alleles, which is twice 160. So all the numbers are working so far. Okay. And then we will go ahead and calculate the proportion of the 2 alleles. So 119 divided into 320, all right, is 0.37, and 210 divided into 320 is 0.62. And you can see that makes sense. 200 is greater than half of this, so it should be greater than 0.5. 119 is clearly less than half of this, so it should be less than 0.5. So use your logic circuits. When you look at numbers from a calculation, just stop a second. Do they make sense? And then our check here, the proportions should add up to 1, and yes, they do. This is all calculated. It's all being calculated here. All right? Then what we do is we calculate the expected frequencies for these numbers here. So we take 160 individuals and we multiply them by 0.37 times 0.37, all right? We take 160 individuals and we multiply them by 2 times 0.37 and 0.62. 2 times 0.37 times 0.62. And that's going to give us 0.467. All right, let's just check our math here real quickly. We'll put a calculator right here. So let's do 2 times 0.37 times 0.63, rounding, equals about 0.46. So you see it's working, right? I left off some digits, so my approximation's not quite the same as here. All mathematicals, calculations, if you knock off decimals at the end, are now an approximation, and we're knocking them off right here. Anyway, we do the same calculation right here. You'll see that 0.62 times 0.62, or sorry, 0.63 times 0.63 will be just a little bit more than 0.39. If I did it correctly, 0.628125 times 0.628125, I should get this number right here exactly. All right, so there's our proportions, and those proportions should add up to 1. Okay, so that's how I calculated this. I simply did p squared. I did 2 times p times q. Well, there's p, and there's q. Here I did q squared, so it's this times this again. There it is. Those are the numbers. And now I'm going to multiply 160 individuals by these proportions. And that gives me an estimated 22, an expected number of individuals that are homozygous for no ALU insertion, 22.12. and 74.74 and 63. So we see the numbers are similar but not identical. And so in lecture I asked you, how different do they have to be before you decide they're different? Or how similar do they have to be before I say they are effectively the same? It's just rounding error or estimating error or things like that. And we would have different answers depending on what was important to us. So we don't want to do that. So the next step, just to complete this story, and then we'll go back to the lecture material, is that's where we're going to use the chi-squared test. So I've gone ahead and set this up so it'll run a chi-squared test right here. All right. And so I've got my observed frequencies. That's 21, 77, and 62 in these three categories of, let me just, I could put them over here, but let's see, minus. That's not what I meant to do. Oh, I forgot. It's taking it as an operator. You have to put a little, and then it'll just display whatever you write. Otherwise, it'll use it as a mathematical argument. So I'll do that, and I'll do plus minus, and I'll do plus plus. There we go. So just to complete the story. And so, here's our observed numbers, 21, 77, and 62. And here's our expected numbers, 22.12, blah, 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 blah. All of those here. And then I'm going to do the chi-squared calculation, which is you take the difference of these, then you square that difference, and then you divide that difference by the expected value. You repeat that here, you repeat that here. I didn't show you all the steps here because the main thing is I wanted to talk about Hardy-Weinberg equilibrium right here. And then we generate a chi-squared statistic. And so we do that for each of the three categories, and then you add those together, and there's this one number right here, which is chi-squared. Okay? And for the record, if 21, 77, and 62 were the observed, and the expecteds were 21, 77, and 62, you would find that this minus this is 0, squared is still 0, divided by 21 is still 0, so this would be 0, this would be 0, this would be 0, and the chi-squared statistic, the total number, would be 0. 0 says the numbers that I'm looking at and comparing to something else, the numbers that I'm looking at and comparing to something else are the same. Well, that's what statistics does. No difference. The bigger this statistic, the greater the difference between these sets of numbers. That's it. That's how this thing works. All right. So, what does that mean? Well, there is a difference between the two. And then the last step on the chi-squared thing is that you look up on the table. So here I've included a published critical values of the chi-squared distribution. I just pulled this off the web. It's easily available. You have copies of it in your reference material for laboratory. And I want to see if this number is big enough to say there is a difference between these things and these things right here. Well, how do I do that? Number of categories minus 1 is your degrees of freedom. So I've got three different categories. Homozygous for one thing, heterozygous, homozygous for the other. In your statistics assignment, you've got something with cards, where there's 4 different suits of cards. Well, that's 4 minus 1, a degrees of freedom of 3, so it would be over here. In our little situation right here, the ALU experiment, degrees of freedom will be 2. It's the number of categories minus 1. That's the rule. Just use it. That, the y is unimportant right now. So we're going to look along this row right here. of 2. And then up here is the probability, the amount of error we're tolerating taking. And remember, these could be converted into percentages. And this represents 5%. This times 100 is 5. This is 5% error. This is 1% error. And this is 0.1% error. This is the typical one we use. So unless you've got some other reason to go somewhere else, let's go ahead and use 5% error everywhere. So our chi-squared critical value is 5.99. And what that means is accepting a 5% error in data collection and number crunching here, we need a chi-squared value of greater than 5.99 to argue statistically that these numbers and these numbers are different from each other. So in this particular case, the number is very small, quite a bit smaller than 5.99. It means there's no difference here. Okay? Let me do something a little bit different. All right? We're still going to go for 160, but I'm going to go ahead and put in 110. All right? And I'm going to put in 14, and the remainder to get to 160, which was our original number. Can't think. 124, so that's 36. 36. There's our 160 again. And we've run through all the calculations. Here's our expecteds, 85 to 62 to 11 versus 110 to 14 to 36. And now look at our chi-squared value. It's 96.69. I'll pull the table up again if I can find it. Here we go. And 96 is quite a bit more than 5.99. Total different interpretation. So this population, with this distribution right here, is nowhere near Hardy-Weinberg equilibrium values. But the previous numbers that we had up there earlier were. So again, you can use this table right here to, come up with any set of numbers that you want and put them in here and run through the calculations and kind of test this whole idea. So that was a long look at a topic. All right, I'm going to pull these out of the way. So that's what we're getting at with the Hardy-Weinberg equilibrium. All right. Okay. So let's talk about some of the things that can affect that Hardy-Weinberg equilibrium. The first one is the population question. So genetic drift is the change in allele frequencies in a population due to random events having nothing to do with the biology of the organisms, eliminating copies of an allele. So right here we've got a small population of brown rabbits, a couple of white rabbits right here. Only 5 of these rabbits reproduce. All right, and notice the genotypes, homozygous for brown, heterozygotes over here. So we create a new population of, rabbits right here. Originally, in this first population, the allele frequency was 0.5 to 0.5, the number of copies of, dominant versus recessive allele right here, or first allele versus second allele. We only took a sample of 5 of them, just randomly picked them and had them reproduce, and notice the rare allele for, pale coloration has still here in this individual. That individual is carrying a copy. These 4 here are all carriers. It's become rarer in the population. It's gone to 0.7 to 0.3. This is what happens in small populations. And then in one more random sampling, it could happen that none of these individuals, so only 2 rabbits reproduce, just these 2 that are homozygous dominant. All of the offspring are now, brown and fixed. And this is the danger of very, very small populations. They genetically do not stay the way they were. You lose rare genes in small populations. And then if, the common genes also happen to include deleterious or damaging genes, that damaging gene is now fixed in the population when it's present in all individuals and there's no alternative. This is the danger of small populations. That's why, it's important that we keep the numbers of rare species up high to avoid this sort of scenario where they lose genetic diversity. Anyway, that's what genetic drift is, and it's the bane of small populations. A similar but related phenomenon is what's called the bottleneck effect. And that is where you have a large population and a whole bunch of them are eliminated for whatever reason, and only a small number are left afterwards. All right? And there's a related version called the founder effect. It's not in here, but that's where you take a large population like the finches back in Ecuador, and you take a very small group, just a random flock of whatever, and, create a new population in a new location. Well, the new small flock may not have all the copies, all the versions of all the alleles for all of the genes that are in this parent population right here. Notice, we're using strong coloration versus intermediate or green here. There's variations in coloration, and we've lost some of that diversity in this surviving population. So here we've reduced a population from very abundant to very rare. That's what happened to buffaloes in North America. Or the founder effect is, again, the finches that landed in the Galapagos were not, were unlikely to have been a full genetic representation of the gene pool of the home population. All right? That's the bottleneck effect. And of course, gene flow is the tendency of individuals to leave one population and move into another population. Emigration from, with an E, leaving a population, emigration out of a population, or immigration into a population. Which, if somebody's emigrating, they're also immigrating over in the other direction. But that's gene flow. So, if something was taking individuals out of the population you were looking at, that's going to alter the results of your Hardy-Weinberg calculation. So this is another one of those assumptions that we don't want to violate. This one has to do with sample size, population size. All right, gene flow right here. All right. Now we're going on to some other ideas that play a role in populations. So for a lot of species, sex is determined by a heritable character, that comes with the parents, right? So males famously, are heterozygous for sex determination. So male mammals, all have X and Y chromosomes, and they determine the gender of the offspring. Females carry only Y chromosomes, so females can only create females, but a female and a male can create a female or a male. Males determine gender in mammals. In some other things, it's the reverse. In the case of this animal right here, crocodiles, alligators, all the crocodilioids, the archosaurs, they are temperature dependent. Gender is determined by temperature, not a heritable thing. So that's a confounding feature right there. If we wanted to do Hardy-Weinberg equilibrium studies of sex ratios, of a population of these animals, we'd have to take the environmental factor into account, which is temperature that the eggs are incubated at. Higher temperature male, lower temperature female, for the record. A number of things do this. All right. Now we move on to another big topic, all right, and this is modes that selection acts upon. Now your textbook, and all your textbooks unfortunately kind of put this over here as just an example of natural selection, but the same idea, the same set of ideas right here applies to artificial selection, and it applies to sexual selection. So all forms of selection, I would just put the word selection up here, can come in one of three forms: stabilizing, directional, or diversifying. All right. And so the deal with stabilizing selection is the population, individuals with either extreme of a character tend to be eliminated, and the mean value of that character is what's rewarded. So that's stabilizing selection. So it favors a mean value, and more individuals fall on that mean value. An example of stabilizing selection in us is birth weights in babies. Babies that are too big, over a certain size right here, have trouble passing through the birth canal. Historically, this was the source of quite a lot of death of mothers during childbirth, and typically the death of the fetus as well because it couldn't get out the birth canal. These days we would do a C-section, so it's not an issue. But if we eliminate medical care, overly large birth weight babies are going to be eliminated. At the same time, very, very small birth weight babies typically are not completely developed. They've been born early. They have not developed at the rate that they're supposed to for a given amount of gestation. There's a couple of different reasons why this could happen. And again, without the presence of substantial corrective medical technology, most of these, historically, these low birth weight babies died off. And so, that is stabilizing selection for a typical weight of somewhere between 6 and 8 pounds using the English system, for birth weights. Okay, that's stabilizing selection. It's always useful to identify cases of this sort of stuff happening in us, but recognize it happens all over the place in biology. Another is directional selection. So this is that, this is the natural selection, red, red fangs, survival of the fittest sort of thing right here. That, this is the sort of stuff that reinforces that idea of natural selection. And that is that, there is a given characteristic in a population. So, coloration is intermediate right here, speckly, sort of intermediately colored right here. But if the environment selects strongly for one extreme of a character, larger, faster, taller, darker, whatever it may be, the whole population over time adapts, to a new environmental characteristic, which is darker backgrounds. And these are the famous melanistic moths of post-industrial England. Normally there's kind of salt and pepper colored with, pale being the defining feature. But when there was coal dust all over everything in England, these moths, the speckled ones became incredibly rare, and what were rare black ones suddenly became incredibly common, and they were the abundant version of this moth. These days England's quite a bit cleaner, not nearly as much soot being released everywhere. They don't burn coal much anymore at all. And the dark melanistic moths are fading away, and the speckled moths are becoming quite common again because they rest on speckled barked trees. And then we finally got and what's an example of this in humans? We're all getting taller. We are taller historically than the bulk of our ancestors. All right, we have less hair than we did back then. There's any number of features in humans that are under directional selection. Our jaws are getting smaller. Let's make the red hump over here. Our jaws are getting smaller over evolutionary time. That's directional selection. Then finally we've got diversifying selection, and that's when the most common individual has the lowest fitness, the lowest likelihood of reproducing effectively. And by the way, the term fitness here is the currency of evolutionary theory. Fitness refers to an individual producing offspring that in turn are capable of having offspring. If you do make a lot of those, you have high fitness. If you don't make any, you have no fitness at all. Anyway, what this says is that individuals that are extremely common have low fitness because they have a high probability of being selected against. Predators fixate on a particular image and, hunt for that image. And if a whole lot of predators fixate on that image, let's say, wolves and hawks that all use their eyes a lot, in searching for prey, they're going to drive these things into, make them very, very rare. And, they're not paying attention to rabbits with dark ears or dark rabbit-shaped things because this is what they recognize and have fixed on as being good food. But when they make it really, really rare, these two versions become more abundant. And what we've done is we've increased the genetic diversity of the population right here by pushing down on this, and making these guys more abundant, And there's lots of examples of this sort of stuff out there. So remember, stabilizing, directional, and diversifying selection apply to natural selection. It's also exactly what we've done in artificial selection of our plants and our animals for food. Directional selection is how we turned the ancestor of corn into the modern gigantic preposterous-looking corn on the cob. The giant modern apple, compared to its ancestor that grew wild on trees and still does in Central Asia. An example from artificial selection. All right. And sexual selection. Also, these three modes right here occur in sexual selection, although there's a great tendency towards directional selection in sexual selection. But in the case of songs of birds, there's stabilizing selection. Songs that are too weird, and too different, in either extreme, let's say, will be selected against, and only songs that are very close to an ideal middle, are responded to in mating rituals of birds. So there's also a thing called frequency-dependent selection, and that's the idea that, you can have individuals that are rare, and I'll go back here, for instance, that are normally rare, like this, what did they call it right here, Himalayan rabbit right here, normally fairly rare, but because it's rare, it escapes predation, it escapes negative effects, and the situation rewards it by it becoming more and more abundant over time. That's frequency-dependent selection, so it can only be selected for, rewarding it with more fitness if it's rare. If it becomes really abundant, and this thing is now rare, the predators are good, they're smart, they're adaptable, they're birds and mammals for, heaven's sake, so they learn. They're going to give up looking for these things because they never find them, and they're going to start identifying these guys as food and select them for a while, and they'll start becoming rarer again, and this thing will become abundant over time. And there's a seesawing back and forth. That's kind of the idea of frequency-dependent selection. In this particular case right here, they're looking at side-blotched lizards and coloration distributions across the population. Examples of sexual selection at work. All right, so sexual dimorphism is typically, there's two reasons for sexual dimorphism. Competition for mates, and that's what's going on with this peacock right here, and that's what's going on with this drake right here of this duck pair. Here's the female, here's the male peacock, here's the peahen, using the proper terminology. And in both cases, there's competition going on right here. In the case of both of the birds, the males have to display like crazy, and the females go, maybe, maybe not. And they'll pick a mate based on something like that. In the world of spiders right here, the size is kind of secondary. But in the world of spiders, the males have to get the attention of the female, convince the female that they're not a threat and that they're not food, and that they're a worthy mate, and then the female may or may not let them mate with each other. In the world of arthropods and small animals, there is often a difference between the male and the female, and almost universally the female will be larger and the male will be smaller. And this is kind of the eggs are expensive, sperm are cheap hypothesis right here. And indeed, eggs are big and occupy a lot of volume, while sperm are very, very small cells and occupy a tiny amount of volume right here. And in this situation, typically the female would live much, much longer than the male as well. So, there's a heavy difference in investment. The female carrying the eggs, the mother of all the new young, is biologically the more important of the two. She's meaningless if there's not a male to fertilize her. But ultimately she, does most of the work of perpetuating the species as far as capturing energy, storing energy, investing the energy in the eggs, protecting the young, making a safe egg nest somewhere, all of that stuff, while the male just inseminates females and gets away if he can. But that's an example of, reproduction having an impact on the physical appearance of organisms, and that's sexual selection. So, here's a little note to y'all. So I wrote this myself right here. So evolution, evolution is a whole bunch of different things, including this idea of natural selection. But don't forget artificial selection, sexual selection in the natural world, and all of these, patterns that we see out there. All right. We talked about this before. Remember that evolution, the change of these species over time, is they're not trying to evolve to become something. The only thing that really drives all life on Earth, all the species around today, everything that's alive today is here because it continued to respond appropriately to the imperative of reproducing. All right? Those things that are not here either, so if anything ever became uninterested in reproduction, biologically for some reason, necessarily they'd go extinct. So only things that reproduce stay around, and that want to reproduce stay around. It's kind of an oxymoron, okay? The other things that are extinct weren't able to adapt fast enough for the changing environment. They don't know what they need. It's just if there's enough genetically based heritable mutations, and the environment is not changing so rapidly that they can't keep up, they will evolve and adapt. If they can't, they go extinct. All right? This is really, really important because, living organisms can't turn into anything, all right? So for instance, tetrapods, things with 4 legs. Vertebrates are tetrapods. We've never seen hexapods in the vertebrate lineage, things with an internal skeleton. There's no 6-legged anythings, no 6-limbed anythings. Just because 6 limbs might be useful, having 2 arms, 2 legs, and 2 wings like an angel, would be really cool. But there's no basis. There's no mechanisms that we're aware of that could generate that in the body plan that is the tetrapod body plan. Mutations show up where vertebrates have extra limbs, but invariably they malfunction and the organism is hampered or outright killed by the presence of that, extra limb or extra pair of limbs. It does not work well, and that's a constraint. And also, there's trade-offs. So let me go back right here. This is what's called runaway sexual selection, the peacock. This tail feather is extraordinary. All male birds have to do the same sort of thing in almost all species. There's a few where the roles are reversed. But very few birds, have this sort of weight of responsibility put on them, this gigantic set of tail feathers right here. And these are expensive. They take a huge amount of energy to maintain. The hypothesis is only the fittest, strongest, genetically super tough, doesn't catch any diseases, resistant to parasites, male bird could possibly sustain this beautiful set of feathers right here, and that's what the female's really, selecting for. But think of it, this bird can barely fly. These guys can't fly. The tail feathers make it almost impossible for them to successfully fly. They might get up into a branch like a chicken, whereas the peahen, these are not heavily flying birds, but she can fly a much greater distance. She's much better at getting away from predators than the male. The male also can't hide very well because he's brightly colored, while she's much more boring. That's typically why females are plain colored in the world of fish, birds, and a heck of a lot of arthropods, including spiders. Their coloration is meant to either hide them or warn predators off, while the male's coloration is primarily, fixed on getting her attention, and so it's got to be loud. And that's what we mean by trade-offs right here. So the peahen, the peacock, has a trade-off in increased, risk of predation, increased cost of his tail in exchange for successful, reproduction. So I want you to review these ideas right here in this chapter. And that is a quick review of the key ideas in Chapter 19.